The overall goal of this procedure is to examine how expression of a gene at different times during differentiation of embryonic stem cells affects hematopoietic differentiation. Following transduction of embryoid body single cell suspension by centrifugation, embryoid bodies are reformed by hanging drop and cultured to allow for further differentiation into hematopoietic cells. Next, single-cell suspensions of the reformed embryoid bodies are prepared, incubated with fluorochrome-conjugated antibodies, and analyzed on a flow cytometer for hematopoietic differentiation. Ultimately, this method demonstrates whether expression of a gene during embryonic stem cell differentiation affects hematopoietic development. It is especially useful for demonstrating roles for genes that may only be active following formation of early embryonic tissue.
So the common techniques to express a gene of interest at different times during embryonic stem cell differentiation require generation of embryonic stem cell lines that inducibly express a gene of interest with inducers such as doxycycline. Generation of these lines can be time-consuming. The advantage of this protocol is that it can be performed immediately, assuming the investigator has the retroviral plasmids for gene expression available.
We first heard this idea about this method when you observed that some genes that we hypothesized would enhance hematopoietic differentiation had an opposite effect in undifferentiated cells. This suggested that mesoderm tissues needed to be formed first before this gene had any effect.
Begin this protocol with